And so it's one of the early biomedical societies and journals in the United States. But as you're about to learn, we're now fully globalized. It's got a very, very rich history. The focus of the society is on the intersection of biomedical research and the practice of medicine. And it has a major focus on mentoring the next generation of researchers. And I hope that we have a lot of students on this video Zoom conference today. The membership costs are extremely reasonable for the society. It's $110 a year for regular members, postdocs $30, and especially reasonable for students where it's free in terms of the membership. So what do you get for membership in the society? There are many perks. I'll mention just a few. You get an annual subscription to the journal, EBM, and we're currently publishing 18 issues per year. Next year, we're going to 24 issues per year. You get free page charges and half color charges for any manuscripts published in the journal, if you're the first or the corresponding author. You can submit abstracts under your SEBM membership to national meetings. We give out travel awards for SEBM student members to these national meetings based upon review of abstracts. And probably most importantly, you're joining a society that will immediately provide global networking opportunities to you. Now I'm going to tell you a little bit more about the journal. I've already told you that we both were established, the Society and Journal, back in 1903. When I took over, which was 15 years ago, we had 7 scientific categories. We now have 20 scientific categories at the crossroads of experimental biology and medicine. We cover the entire translational spectrum from T0 to T4. We've globalized the journal. We've got offices now on 5 continents. We've got, in addition to myself in the United States, global editors in Taiwan, Great Britain, Brazil, and Ghana. And we have a China outreach office in Chengdu as well. The editorial board is clearly international with the 20 associate editors and over 160 editorial board members coming from all over the world. Our current 2-year impact factor is above 3. And I guess the last thing I'll mention is that every year, or almost every year, we had to skip this year because of the pandemic, we run an International Experimental Biology and Medicine Conference, IEBMC. The one that will take place in October 2021, hopefully the pandemic will be over by then, is going to be focused on regenerative medicine, and it's going to take place in my own town, Memphis, Tennessee. So now I'll introduce the speakers. The speakers are Hana El-Samad, who is a professor of biochemistry and biophysics at UCF School of Medicine. Her laboratory focuses on fundamental questions to uncover the basic design principles of biologic signaling, with a focus on inter and intracellular communication and processing of single and multicellular systems. They also are focused on evolutionary rewiring of conserved pathways across multiple species and population heterogeneity in cellular decision-making. And kindly stepping in for Mike Jewett, we very much appreciate that our other speaker is Ashti Karim, who is a research assistant professor at the Center for Synthetic Biology at Northwestern University. Ashti is developing cell-free systems for biosynthetic pathway prototyping and natural product discovery. And I think, since I don't see Hana yet, our first speaker is going to be Ashley. And so I'm going to hand this over to Ashley, and then we'll go directly into Hannah's presentation, if she is on in time.
Awesome. Thank you, Steve. And it's really my pleasure to be here. It's wonderful that this seminar series is going on to introduce synthetic biology from many different angles. For me, it's wonderful that Mike Jewett and I get to work together with a talented group of students and postdocs, all with the goal of really engineering the process of protein translation for many different purposes, from generating non-natural polymers to informing cellular function to manufacturing therapeutics and chemicals. We have many different projects. Our lab is spread across engineering the translation system to facilitate the ribosome catalyzing new chemistries, non-proteinogenic chemistries, to programming life. The idea, can we use cell-free systems to prototype transcription and translation to better inform cellular design, as well as developing platforms for on-demand biomanufacturing of glycoprotein therapeutics and vaccines, the production of sustainable chemicals, using cell-free systems to develop diagnostics and sensors in real time. And most recently, we've expanded our research efforts towards developing educational kits to teach students how biological concepts occur in real laboratory experiments. And so today I'm not going to talk to you about all of these things, but I'm going to focus on informing cellular design with a specific angle at looking at sustainable chemical production. And so my title here is, how are we using cell-free systems basically to accelerate industrial biotechnology? This is a collaboration with the company LanzaTech that exists just outside of the Chicago area, down the street from Northwestern here. And what I hope I'm able to show you today is how we're using cell-free approaches to be a powerful complement to traditional metabolic engineering efforts. So nature produces many valuable chemicals, from artemisinin, which is a anti-malarial drug, to paclitaxel, an anti-cancer drug, to one of our most valuable chemicals, ethanol. And nature produces many other diverse chemicals as well that are vital to our health, from neurotransmitters to immunosuppressants to antibiotics. And this is shown, I would say, more clearly by this graph here, which shows the percent of all new small molecule drugs that are FDA approved that are derived from natural products or are natural products themselves that have been approved over the last 40 years. And you can see that nearly 30% of all approved small molecule drugs come from natural products. This is not a small number. The problem, though, is that natural sources, like the plants I just showed you, are often insufficient to meet societal demand. Luckily, as engineers and scientists, we've gotten really good at harnessing model microorganisms like E. coli and yeasts to take up simple starting substrates like sugars and convert them to a wide variety of valuable chemicals, from simple chemicals like isopropanol to muconic acid to even more complex molecules like artemisinin. And all of these molecules here are produced commercially from biological sources. The real problem that we get to, though, is if we look back here, many of these organisms are only good at taking up sugars, and the number of feedstocks that they're able to convert into these chemicals are limited. And while many researchers are trying to adapt these organisms to take up feedstocks that would be more conducive to increasing demands to fight climate change, as well as trying to reduce our reliance on fossil fuels, LanzaTech are experts in harnessing this organism here. This is Clostridium autoethanogenum. It's an obligate anaerobe And what's nice about this organism is it can take a wide variety of feedstock waste gases, such as industrial waste gas from steel facilities, to biogas, to solid waste that's been gasified, biomass, and even carbon dioxide directly. And it can take clean or dirty streams of these gases and convert them very efficiently to ethanol. And actually, LanzaTech has built a commercial plant in China that's retrofitted on a steel mill currently that's making millions of tons of ethanol per day. So this is a reality that LanzaTech is pursuing, and it's their approach to green chemistry for the future. The problem is this organism, while it can take up many different types of carbon waste streams, it doesn't have access to this wide palette of chemicals. And it's actually really difficult to get to the point where this organism could be as great as some of our model microorganisms. And the reality, we can see this better when we look at how we might engineer one of these organisms. So let's look at a model microorganism. Let's say we want to make butanol, which is a potential alternative fuel and precursor to other chemicals. If we wanted to make this in E. coli, we could make it from glucose, harnessing native metabolism, such as glycolysis, to get to an intermediate acetyl-CoA. From acetyl-CoA, we can define the chemical reaction steps that would get us to butanol. We can then identify enzymes that perform these reactions, and we can identify different homologs for each of these reaction steps using online databases that curate enzymes that perform particular functions. Once we've identified a biosynthetic pathway that we'd like to put in our organism, we can then build it by using modern molecular biology techniques where we can take DNA encoding these enzymes and put them in our model microorganism. Now, if you want to put 5 different enzymes inside of an organism here, this may take some fine tuning and strain optimization, where we're designing, building, and getting to the point where we could test this organism to see if it makes enough butanol. I don't know about you, but in my hands or my students' hands, testing only a handful like this would take about a month and a half to do, all to find out that we're not making butanol at the titers or rates that we need them to. So we have to go back to the drawing board, designing, building, and testing for this phenotype of interest. Let's say we just wanted to test 5 enzyme homologs or variants for each of these steps. All of the simple possible combinations that we could get to equate to over 3,000 combinations. And this is a number that's very high, but that's not even considering if we wanted to tune the levels of these enzymes and how they're expressed inside of our microorganism. And so if we wanted to test just 3 enzyme levels for each of these and do every possible combination, we're getting to well over 7,000 possible combinations. And this just exceeds typical pipelines today. And you might argue that some companies are getting to these numbers, but that's with model microorganisms like E. coli and yeast. And when you go into an organism like Clostridium, that's an obligate anaerobe, these numbers are just not achievable. The reality is that design-build test cycles for microbial biochemical production are time-consuming, and it's essential that we speed up these processes so that we can get access to many different biochemicals. One way that we speed up this process, and we've done for many decades now, is to use purified single enzymes, study them in vitro, and learn about different enzyme kinetics that then allow us to select enzymes to put inside of our organism. The problem with this is just by selecting the best enzymes that performed well in vitro often don't perform well when you put it in the milieu of all of metabolism inside of a cell. And so in the Jewett Lab, we believe that one way to bridge this gap is to use multi-enzymatic cell-free systems. The way this works is that we take E. coli cells, we grow them up, we break them open and remove cellular debris and genomic DNA. And what we're left with are crude cell lysates that, that contain the machinery for transcription and translation, enzymes for native metabolism. And what we can do is we can add DNA encoding all of the enzymes that we have of interest that we'd like to test, perform in vitro protein synthesis, transcription and translation to enrich each of these lysates individually with the enzymes that we'd like to test. We can then combinatorially mix and match these enzymes, like making a cocktail from different ingredients. We can then add substrates and observe product formation to be able to test different biosynthetic pathways. In this way, our design build test unit is now an enriched lysate containing enzymes of interest rather than an organism or DNA. We can then, let's say we wanted to make butanol, we can express each of our enzymes for the butanol pathway, all 5, mix these 5 lysates together, add glucose, salts, and cofactors, and after 24 hours of incubation, we can produce butanol in this system. By using the cell-free approach, we can decrease the time it takes to build pathways to a matter of a day, and we can increase the throughput because now we can operate in a tube or even in plates. This approach, which we've called in vitro prototyping and rapid optimization of biosynthetic enzymes, or iPro, is 20 times faster to build pathways and has a 2 orders of magnitude increase in throughput over cellular systems. But the real question comes back to, okay, we have this cell-free system that now allows us to test lots of biosynthetic pathways. How do we relate that to our cellular systems? Well, we know that they don't equal each other. We've known that from previous in vitro experiments over the last 50 or so years. But we know they're not totally unequal. These are useful to inform our cellular experiments. So to answer this question and to define this relationship, we look at our cellular systems and what are the metrics important to say this cellular system is good for producing our biochemical. Those metrics are titer, rate, and yield. Are we achieving those? And enzyme expression. We don't want to select enzymes that don't express well inside of our cells, whether they're insoluble or just don't express a lot of that enzyme. These are metrics that we also have in our cell-free systems. So how do we use this information to develop a correlation between the two? To show you how we've begun to do this, let's look at a simpler example than butanol production, the production of 3-hydroxybutyrate from acetyl-CoA. So we're using native metabolism from glucose to get here. And this requires 2 additional enzymes besides what's already present in the lysate. What we're going to do is design 5 biosynthetic pathways where we keep the level of this HBD enzyme constant and we individually titrate the amount of this THL enzyme that we have. We can then build this by expressing each of our enzymes and then combinatorially mixing the titrated amount of THL here to produce 5 different combinations. We can then observe 3-hydroxybutyrate production over time here. And what you can see is this is just 5 designs, and we have a lot of information embedded here. And it's hard for me to say whether this pathway is better than this pathway. So it would be nice to have a single metric that does this. So let's look at what we actually have here. In these 5 designs, we have a titer. So when the reaction is complete, how much 3-hydroxybutyrate have we made? We have a rate of production during the most linear or most productive regime. We also have some measurement of our soluble yield, as well as how much enzyme we're adding to our system. So this penalizes for having too much enzyme. In a cell-free system, I could add millimolar or molar quantities of an enzyme, but that wouldn't be useful to informing cellular design because there's no way that I could actually get that amount inside of a cell. So for having too much enzyme should penalize a score if we're trying to create one here. So we can add these 2 together, the solubility factor as well as the total enzyme concentration, to get some metric for our enzyme expression. We can then multiply our titer by our rate, by our enzyme expression, and what we're left with is the tree score, which is a single quantitative metric that exaggerates differences in titer, rate, and incorporates some metric of enzyme expression. This will allow us to test many more pathway designs in cell-free and quickly screen for higher and lower performing pathways. So to give you an example, let's look at 3-hydroxybutyrate production. We're going to test 6 enzyme homologs for each of these 2 steps. We're going to try all the simple possible combinations. So we do this by expressing each of our enzyme homologs by cell-free protein synthesis, and then mixing them to create 36 pathway combinations. We then measure 3-hydroxybutyrate production and calculate tree scores for each of these. And what you can see is that only a handful actually produce quantities of 3-hydroxybutyrate, and many of them don't. And so what we wanted to do was take our highest performing pathway in cell-free, our lowest performing pathway, and 2 that fell in the middle, and build those in Clostridium. So we handed these designs over to LanzaTech, and they built each of these 4 designs. And over the course of a fermentation on carbon gas, they were able to measure 3-hydroxybutyrate production. Their highest performing pathway was also our highest performing. Their lowest performing pathway, A here, was also our lowest. And the other 2 fell in the middle. And so what this tells me is that iProbe is useful in selecting or down selecting pathways that don't perform well. And I would argue that B, C, and D are all pathways worth pursuing in cells. LanzaTech only had to produce 4 strains testing these pathways rather than 36 combinations, which would have taken months to build. So what we've done from here is we've taken this iProbe selected pathway and we've scaled up these into larger fermenters and optimized fermentation conditions to get to a point where over the course of a 2-week-long fermentation, we can produce 3-hydroxybutyrate at nearly 150 millimolar without significant defects in growth. And so this continuous carbon gas to 3-HB system produces 15 grams per liter with no of 3-hydroxybutyrate with no additional genomic modifications. This is the highest reported titer of 3-HB and leaves room for LanzaTech to continue modifying their strain to improve the production rates here. Their efforts can be focused now on the actual metabolic engineering of their strain versus selecting a pathway for their molecule of interest. And so, not to belabor the point, but I want to emphasize that this really sped up our design efforts significantly. And so if we look at what I just showed you, which is this cell-free approach versus doing everything in Clostridium over the 3 rounds of design build tests that I just showed you. In our first round, it took us about a month to get the DNA and to perform our cell-free experiments. It would have taken us 4.5 months to do this in Clostridium. If we went through a second round after analyzing that data, this increases it more. And our third round, we get to, this would have taken us about a year in Clostridium, where it only took us a month and a half. And I know what you're thinking, that's not fair because we ended up putting it in Clostridia to test whether this was a worthwhile prototyping effort and get to our final strain. So when we do that, we get to about 5 months. So the longest time here is the stage at building these strains because of how long they take to grow and how hard they are to manipulate. And so this really reduced the time it took to build these strains. So now let me move on to the production of butanol, what I started with. So it's a longer pathway. Let's apply the same iProbe approach to testing these. So if we look at this pathway, we're going to test 6 different homologs for each of the steps here. So if we do all of the simple possible combinations, we're already getting into over 1,000 combinations. And if we wanted to test different enzyme levels, which we have seen to show a significant impact on pathway performance, we're starting to get to numbers that are even difficult to test in our cell-free systems. Where do we start? And so this will require iterations of design build test efforts. And what we've done is we've coupled with Lockheed Martin, who are experts at using computational approaches to design materials, to apply some of those computational efforts at designing biosynthetic pathways. So using a data-driven design of experiments approach, we use computational neural network-based algorithms to predict what pathways should we start testing. And so we built those by expressing each of our enzyme homologs via cell-free protein synthesis, and then mixing our pathways as I've shown you before. Over the course of 5 iterations of design build tests, we were able to increase butanol production 4-fold in our cell-free systems, and testing over 200 combinations in a matter of 12 days that it took to build these pathways. And so what we did from this is we further analyzed this to glean information that we could then use to build cellular systems. So here I'm showing you our iProbe tree score on the x-axis and the Clostridium production of 3-hydroxybutyrate in gray, which I already showed you, and then our butanol producing strains in black here. And what you can see is there's a positive correlation between our cell-free experiments and our cellular pathways. This was surprising to us. We were hoping for this, but at the same time, we were using E. coli-based cell-free systems to screen pathways to ultimately inform expression in Clostridium. All of our biases led us to believe that this wouldn't work as well as what we see here. And so while a Clostridium-based cell-free system would likely improve our prototyping efforts, this right here demonstrates the power of coupling data-driven design with cell-free prototyping to select feasible subsets of pathways worth testing in cells. And so what I hope I've been able to show you is that we've developed a cell-free approach that's much faster to build pathways and allows us to access a higher degree of throughput. It can't be overstated that we can now select and optimize pathway designs without having to re-engineer DNA or organisms. So we can take pathways of interest designs, test them in cell-free, port them over to cells, and scale them up in order to reach the chemicals that we'd like to make. And so this cell-free frameworks ideally sits coupled with LanzaTech's Clostridia BioFoundry, with computational design efforts and modeling approaches, underlined by omics analyses that allow us to get to biochemicals much more quickly. Ideally to expand this chemical palette available to Clostridium. And so lastly, I'd like to thank my partner here, Mike Jewett, and all of the students and postdocs that have worked with us, along with Lockheed Martin, LanzaTech, and our funding sources. So I'd like to take any questions, or I guess at the end I'll take questions, but thank you for listening.
So Ashley, thank you. That was terrific. Hannah has not joined us as of yet, so I think we should go directly into the question period, and I'm handing that off to Caitlin Hart.
So for anyone on Facebook Live, you're welcome to type questions into the comment section, and we can relay those. Or you can use the Q&A or the chat feature in Zoom to relay any questions to the speaker today.
Can't hear you, Caitlin.
Is this better if I hold my mic up?
No.
How about now? Okay. Is this better?
It's very quiet. We can hear, but it's very quiet.
I will try my best to talk loudly. Does this help?
That's a little bit better.
Okay, I'm so sorry. I'm not sure what's going on. So one question I do have, and this is possibly just my inexperience, but in general, with making these enzymes and purifying them, how tedious is that? I mean, is this something that's pretty mainstream and easy to do, or is that potentially a hang up at times?
So what's unique about these systems is we use, I showed you, or briefly mentioned how we grow up E. coli, break them open, and what we're left with are the crude lysate. And so in many in vitro-based systems, you have to deal with enzyme expression, usually in a host organism, that you then purify after the fact, right? In these systems, we're not purifying anything. And so directly in the crude lysate that has a lot of enzymes, small molecules, et cetera, we're producing the enzymes using transcription and translation. So we take that lysate, add the DNA encoding our enzyme. In vitro, we're doing transcription and translation. The protein is made, and then we don't purify that afterwards. We then take that enriched lysate now and mix those with enriched lysates that have each of the enzymes we'd like to test. And now we have sort of a cocktail or a mixture that has E. coli-based lysate, as well as our what would be heterologous enzyme that we're expressing them outside of the cell. And so no purification needs to be done. In this way, we can kind of use a generic E. coli strain that we make a bunch of lysate out of, and then we can sort of do all of these prototyping efforts from that same lysate.
So Caitlin, would you like, it was very hard to hear your question. Would you like me to ask the question that I see from Peter Stambrook?
Yeah, if you're still having troubles hearing me.
Okay.
I apologize.
That's okay. So Peter Stambrook has the following question. Since you are adding to cell-free systems, won't you be getting a large number of unwanted side products?
Yes, is the short answer. And while normally you might consider that a negative, for us, it's a positive in one way. If we're trying to prototype in cell-free for a cellular system, what we call this sort of byproduct formation is native-like metabolism. So it's not exactly the same as metabolism. Metabolism is dynamic inside of a cell. That's not occurring the same way in our cell-free systems, but because we have all of this extra stuff that's creating byproducts and diverting flux of the carbon that we'd like to go to our product, we can get a better idea of how it might perform in a cell. That pathway might perform in a cell. Versus a purely in vitro purified system, we'd get probably a really efficient pathway, but that might not resemble the cellular system as well. The caveat here is we still are in E. coli cell-free systems looking at Clostridium. So that's one of the reasons why we found it very interesting that we found a correlation, and we're continuing to develop that correlation. But it would also be useful for E. coli-based systems. Can't hear you, Steve.
Are there any other questions that anybody has? Jess, are there any questions from Facebook?
I'm not seeing anything, if you can hear me.
Okay. So since Hannah has not arrived, I think we're going to give everybody back a little bit of their time this afternoon. And I want to again thank Ashley. I really enjoyed your presentation and I learned a great deal from it. So thank you very much.
While it might not be the exact fit for the journal or the audience, I hope I did teach you a little something about what we're doing with synthetic biology with the shared common goal of trying to improve the world. So I hope you enjoyed it.
So it's a great fit for the journal. Keep that in mind. Thank you very much. So long, everybody.
Take care, everyone.